Okay. So nuclear division comes in two forms. So you've either got, and we're going to look in detail at only the first one, mitosis. We don't need to know the details of what happens. In fact, other than this definition of meiosis and then the results of meiosis, we don't need to know an awful lot more. Sorry, the results as in what it's used for, what meiosis is used for. So let me just explain. Sorry, just got my settings messed up here. Okay, back on track. We are we're going to look at mitosis in detail. If you're asked for a definition of mitosis, it's something that's worth really checking yourself regularly on to make sure that there's quite a few parts of it that you need to make sure you have nailed down. So the first top of the list of things you need to say about mitosis, if you're asked what's meant by mitosis, is you need to say that it's nuclear division. Okay, so whether you say it's the division of a nucleus or it's a form of nuclear division, whatever you say, you need to make that point. That's your first key point. Okay, because it's not cell division. It's just, it's strictly speaking, it's how nuclei divide. And actually, here's the thing, later on in the course, you'll see, and you might have seen it already with for example, in plant reproduction, sexual reproduction in plants, you can often have cells in which mitosis has happened and the cells don't split at all. In fact, you can have multiple rounds of mitosis happening within one cell where the nuclei divides, and the nuclei divides again, and even again, you end up with like these bunch of nuclei inside one cell. So they are actually really separate kind of ideas, right? Mitosis is usually a precursor and usually the kind of, it's the way or part of the way that cells are able to finally split in two. It's the reason why they're doing mitosis is so that they can split in two, but it's not always the case. So the division of a nucleus is what mitosis and meiosis is actually, okay? But the difference comes when you go further looking at mitosis. The difference between mitosis and meiosis comes in the sense that mitosis is about the production of two from one cell, right? The production of two identical daughter nuclei. And the key thing about those two daughter nuclei is that they are not only are they identical to each other, but they are genetically identical to the original mother cell. Now, where's the original mother cell? Well, it's gone now because it just split in two, so there is no original mother cell. Those two daughters, though, and this is the key point about mitosis, they are genetically identical to each other and to the original mother cell. Not so with meiosis. In meiosis, not only it's nuclear division, but we'll see in a second it can only happen for diploid nuclei. Mitosis can happen with either diploid or haploid nuclei. It's not a problem. But with meiosis is restricted. It's a very special case of nuclear division. It can only work with diploid nuclei, and the reason is because the daughters that are produced, there are four of them, first of all. They are all genetically different from each other and from the original mother cell, not least of which because they're haploid. They only have half the number of chromosomes compared to the original mother cell. So that's a big difference to begin with. But they're also going to be genetically different to one another. All these daughters are going to be genetically unique, right, to each other. But they are also, this is the reason, rather, that meiosis can only happen with diploid nuclei, right? Because if you're going to have that the daughters are going to have half the number of chromosomes compared to the parent, well, the minimum amount of chromosomes you can have is the haploid number, whatever that is. For in humans, it's 23, right? You couldn't have a haploid 23 chromosome cell from a human, let's say a gamete, right? You can't divide gametes, or gametes can't be divided by meiosis because they have 23 chromosomes. So if you were to split that so that each daughter ends up with half that number of chromosomes, you immediately run into trouble. The maths just don't work because you're ending up with 11 and a half chromosomes. They don't do half chromosomes. Cells don't. So that's not going to work. So the only way meiosis happens is with diploid nuclei. Because diploid nuclei have two sets of chromosomes, each of the daughters can end up with one full set. Okay? Then we get cytokinesis. So interphase, followed by some sort of nuclear division, followed by cell division. So the once the nuclear division is complete, generally speaking, what happens then is the cell splits in two and you get this cytokinesis event. And we'll look at that in detail later on, but where we want to look at is what happens with the details of mitosis, right? We'll go back to that in a second. This is a pair of homologous chromosomes I was talking about. These are not sister chromatids, okay? These are individual chromosomes. One came from one parent, one came from another parent. These are the pairs of chromosomes we talk about in a diploid cell. We've got 23 pairs in humans. Each of these structures, right, there's two structures there, two chromosomes. Each of those will end up being fully replicated and have a sister chromatid attached to it, right, to turn it into one of these X-shaped things. So this is a pair of homologous chromosomes for which that hasn't happened yet. There hasn't been a DNA replication event. It's just a picture, right? But what you'll notice is these are along, that little dent there, that's the centromere. That's the position or the structure where another sister chromatids will be attached to another. And it's also, as we'll see, where the spindle fiber is attached during mitosis. But these other bits are just highlighting where genes might be, right? The positions on the chromosome of a particular gene. So, you know, a particular gene for whatever it is, eye color, hair color, how your heart muscles are working, you know, what enzyme exactly you're going to produce, what hormones are made. All of these are coded for by sections of DNA that we call genes. And they have particular positions along these chromosomes where you'll find them. And in humans, for example, you know, on a particular chromosome, you're going to find the enzyme for, you know, catalase or something. You're going to find that at position 1498 on chromosome 23, right? That's the way, that's the kind of thing we're looking at. And we'll look at the other stuff in more detail later on. I said at the beginning, I did say this topic is not very kind of, you know, user-friendly because of just the language. Look at all these C words. It's just really annoying. And some of you will have learned even other associated words that I'm almost even afraid to say, but I'll say them in a little while. I'll mention them in a little while as words that we don't really need to know as part of our story. But chromosome, we've talked about. Chromatin is the loose form. Chromatids then is one of the two identical copies of DNA that are held together at the centromere, and the centromere is the location or the position or the place on the chromosome where the two sister chromatids are attached.